So last week we introduced several pump, P-class pump, V-class proton pump, and the F-class proton pump. They only transport ion, okay? But now let us move on to the ABC superfamily. It can pump a lot of different stuff. Of course, it can also transport ion, but it can also transport larger molecule like amino acids, sugar, peptide, lipid, and many, many other small molecules, okay? So ABC superfamily, ABC stands for ATP binding cassette, okay? Because all the members in this family contain the ATP binding cassette. And they are transmembrane protein. They have two transmembrane domains, okay? So those ATP binding is really important to change their conformation and also important for them to transport different molecules. So why they can transport different molecules? Because as you can see here, this is the protein structure of the ABC members. So the blue part show the hydrophilic amino acid, and the yellow part is hydrophobic amino acids. Okay, and they have such a very special structure called a cavity. Such a cavity extends halfway to the membrane. Okay, so ATP binding will change this conformation from this and to this. They will keep changing between these two conformations. So the molecule will have either hydrophilic or hydrophobic molecule will have affinity with those amino acids. They can kind of extend to this side, okay? And such affinity is not a 100% binding. You need to know the affinity is weak enough and strong enough. They always like associate and the deassociate keep shuttling between these two status, okay? So they will bind and dissociate, bind and dissociate. So by using the energy from the ATP hydrolysis, they will change the conformation between this and this. So they will face to the other side, and those molecules, well, with such a conformation change, they will release and go to the other side. Okay. By doing this, they can kind of pumping molecule from the one side of the membrane to the other side across the membrane. Okay. So this table summarizes the different members of the ABC superfamilies, and they have a different function, and the defect of those protein will cause several different diseases, okay? And actually, originally, in early days, they don't call ABC superfamily. They call the MDR1, MDR, okay, which stands for multiple drug resistant transport protein. Why they call this name? Because when people try to use some toxic, okay, some chemical, those are toxic, try to kill the cancer cell for the chemotherapy. They treat those toxins, but they found those toxins can easily kill normal cell, but cannot kill the cancer cell. Okay, and finally they found why those toxins cannot kill the cancer cell, because the cancer cell has a lot of this kind of protein that keep pumping out those toxins to the extracellular space. So those toxins will not retain inside the cell. That is why this protein will help those cancer cells to achieve so-called multi-drug resistant ability. So they originally named multi-drug resistant transport protein. And finally, after more and more research, people know all of these members contain the ATP binding cassette. They share the same mechanism to pump a molecule outside the cell. So they renamed this to the ABC superfamily. Okay? And I would like you to notice this, the ABC before, which the flip path we introduced last week. Okay, so the last week we discussed about this, introduced this, the phospholipid synthesis is a multiple step reaction. It will synthesize in the cytosol first, then it will, those intermediate metabolites will go to the surface of the ER membrane, and the several key enzymes on the ER membrane will kind of in charge of the following reaction. And the PC, okay, in the beginning, though it's originally during the early stage of the synthesis, it located to this leaflet face to the cytosol. But this flip path helped them to flip to the other side. So this flip path is ABC before. It can help use ATP energy from the ATP hydrolysis to flip this phospholipid to the other side. Okay, so this is the guys who determine the lipid so-called asymmetric. Okay, and how to next textbook just give you an example to show you how to do the research or how to do the experiment to prove this ABCB4 have a flipping activity. So they use this experiment to prove this. Okay, so first they will kind of isolate the vesicle contain ABCB4. Okay, basically those ABCB4 they will locate to secretory vesicle. So those vesicles will gradually secrete out to the median. So you just collect median and use ABCB4 antibody, which will bind to ABCB4. So those antibody will also conjugate with some agarose or some beads. They will align to the column wall of the column. So you just go through those median and the final several vesicle content ABCB4 will bind with your antibody connect to the wall. Okay, so you will collect a lot of ABCB4 containing vesicle. Okay, so next you just add, after you're collecting those ABCB4 positive vesicle, you just add a fluorescent phospholipid. Okay, those have a fluorescent signal can be visualized by some kind of machine. Okay, so those lipid of hydrophobic part will align with the hydrophilic part will align with hydrophilic. So after a while, those fluorescent phospholipid will incorporate into the membrane structure. Okay, then you separate your reaction to the two groups. The first group, you don't add anything. You don't add ATP. You don't give them energy. But the other group, you add ATP. So with ATP, theoretically, ABCB4 can use this as energy to flip a molecule from this leaf lab to the other leaf lab. Okay, but without the ATP, they cannot do anything. They can do nothing. Okay, so next will be the key point. You just add the quencher, which is a chemicals. Okay, will make a fluorescence dimmer. We call it quencher. Okay, and this quencher is a membrane impermeable quenching compound. They only can affect molecule outside on the surface. They cannot permeable to and affect anything in the inner membrane leaf lab. Okay, so after adding this quencher, it would make the phospholipid, fluorescent phospholipid on the surface become dimmer. Okay, and also affect this the surface of phospholipid, fluorescent phospholipid will become dimmer. Okay, but if ABCB4 can really function to flip the phospholipid to the inner leaf lab, they can protect more fluorescent phospholipid from the quenching reaction. Okay, so if you can keep more fluorescent phospholipid from its quenching, it means this ABCB4 flipping activity is higher. Okay. So that is why in the beginning you just measure after incubation those vesicle with the fluorescent phospholipid, you will get the basal level of your fluorescent signal in the beginning. Then you just separate two groups. One group is a black light curve. The other group is showed as a red curve. You just add ATP or no ATP. Okay, and adding quencher, you will see with the group with ATP can retain more fluorescence signal compared to the group without ATP. Okay, so those are different come from the activity of the ABCB4. They deliver more of fluorescent phospholipid to the interior leaf lab. Okay, but after that, you need to do the one more kind of positive control, like add detergent, because what you observe is all number. Okay, you only see the number here and it declined here. You need to make sure those signal, what you are observed is really come from something about the membrane structure. It's not like a debris or some garbage. Okay, you need to make sure those signal is really come from membrane-based structure. That is why you add detergent. Detergent will destroy any membrane-based structure. Then you see the signal decline at all. So anything above this signal it's all come from the membrane-based structure. So you know those signal, kind of confirm those signal, what you are measure is all based on the membrane structure here, okay? That is the way people can use to prove that ABCB4 can really have a flipping activity. With ATP hydrolysis of the energy, they can flip more phospholipid to the other leaf left. Okay? Any questions so far? No? Okay. So, and the other member which is important in the ABC family is CFTR, which stands for cystic fibrosis transmembrane regulator. Okay? So this, as you can see here, is also contained two ATP binding cassette in the true transmembrane domain, but they are not the pumping protein. Okay, they cannot actively transport molecules. Okay, they are kind of chloride channel, not a pump. Okay, so the ATP binding so ATP will bind here, then the second ATP will bind here, which ATP hydrolysis would help them to change the conformation and just open the channel. Okay, but the transportation is totally driven by the electrochemical gradient of this chloride. Okay, they don't drive the transportation. Such energy only help them to open the channel. Okay, so the energy is just indirectly facilitate the chloride delivery. Okay, but after ATP hydrolysis, it will go back to this conformation, and they will close the conformation of those CFTR. Okay, and CFTR is really important. Why is so important? Because it's directly cause one kind of disease called cystic fibrosis. Okay, so this is a genetic disease which will kind of cannot characterized by abnormal fluid secretion from the tissue and caused by the defective chloride channel. So in our airway, as I mentioned before, there on the surface of our airway, there are a lot of multicilia. They will keep bending and generate such a flow. It can help us to take out or remove the mucus. Okay, so there will be less bacteria or debris retained in our airways, so we can reduce the inflammation. Okay, but to help them to kind of easily bend and generate this flow, you need to hydrate this environment. Okay, so the chloride, not only water, you need to also add some chloride, some ion, which can hydrate such a solution, which will make more wet of those environments. Okay, so the CFTR on the surface of the airway epithelial cell, which will be the key molecule to deliver the chloride and hydrate the mucosa layer. So with such a environment, the multicilia, they can bend and generate such a flow. But the mutant of this CFTR, those CFTR cannot locate onto the membrane of those epithelial cell. They cannot deliver the chloride. So the water cannot retain in the airways environment. So the whole airways will be really viscous, really sticky. So they cannot bend. Okay, too sticky, they cannot bend. So a lot of bacteria or debris will retain in our airway, and which will trigger really severe inflammation and finally cause a lot of different diseases. Okay, so the one channel defect will cause a lot of different diseases. Okay, that is CFTR. So you need to remember this mechanism, how CFTR function and why the defect of CFTR will cause cystic fibrosis. Okay. Okay, so we just really quickly give you the example how the ABC superfamily flip the lipid and also transport some ion. Yeah, any questions so far? No? Okay. So next, let us talk about this. So we talk about the membrane potential. Okay, so what is membrane potential? Membrane potential is a voltage different across the membrane. Okay, but if there's across this stuff, if these two pools do not have any voltage different, there will be no membrane potential. But if these guys can transport a lot of sodium from this side to the other side. I mean, cation to the other side. They will accumulate more cation here and more anion here. Okay. In this case, they will be kind of have a voltage different. Then this system will look like a battery and have a membrane potential. Okay, because more cation would like to go back here from this side to the other side, which will generate such a potential. Okay, and also this will be also the case if there are some potassium channels which can deliver potassium from this side to the other side. They will accumulate more cation here and more anion here. Then will also generate the membrane potential. This is also the case of the plasma membrane. Okay, the ion cation, the distribution of the cation and an ion on our plasma membrane is asymmetric. Okay, there are more cation on the outside of cell and more anion inside the cells. The cell in our body are negative charge. Okay, so why they will generate such a membrane potential? That's because there are a lot of different channel will release such. There are a lot of different ion channel on the plasma membrane, especially the potassium channel. There's a one channel called the resting potassium channel. It's not regulated by the voltage or any stimulation. They keep open on the steady state. So a lot of potassium will keep a release to the extracellular space. That is why even in the resting state, they keep open. So a lot of potassium will release to the surface, then accumulate more cation on the surface. Okay, then and generate such a membrane potential. So next, we want to know, when we talk about this ion channel, we will say this is a potassium ion channel, this is a sodium ion channel, this is chloride ion channel, right? Why would they call the potassium ion channel? Because they can only allow the potassium to deliver across the channel, right? But how such a one kind of protein can achieve the specificity? Why it only can transport potassium, but cannot transport other ion? That's weird, right? How can they really tell? Okay, so next we will use this example to show you why the resting potassium channel only transport potassium but not other cation. Okay, so because this is a well-studied the channel, so we use this as an example. The people found this already got Nobel Prize. Okay, so this is the monomer of the resting potassium channel. And as you can see here, there will be two alpha helix, which are their transmembrane domain. And you will see this part is called a P segment. Okay, the red part is a P segment. So this is the this is the domain which can contribute, which is required for, is critical for such a ion specificity. Okay. So basically, this is a monomer of the resting potassium channel. Four of the resting potassium channel were formed one kind of real channel here. They work together. Okay, it's a tetramer. Okay, so this is the top view of those tetramer of a resting potassium channel. Their P segment will face to the core and the core of the channel here. Okay. And as you know, those cation or anion in the aqueous solution, they have charge. They would like to bind to other charged molecule. Like an ion molecule will bind to a cation molecule due to the charge, right? So in the aqueous solution, for example, those are potassium, they would like to bind to the other polar or charged molecule. So although the water molecule don't have charge, they don't have net charge, but they are polar molecule. They are more negative in this side and more positive in this side. So potassium in the aqueous solution or in the water solution, their negative side of the water molecule will bind to the potassium. It's also the case for the sodium ion. A lot of water molecule will bind to those sodium ion, which is an energetic favorable condition. Okay, but potassium, although they are both positive charge, potassium ion is a little bit larger than the sodium ion. Okay, So the surface of the potassium is good for the 8 water molecules, but the sodium ion is only good for the 6 water molecules. So the potassium ion is slightly bigger than sodium ion, okay? And the amazing part is, so binding with an 8 water molecule is, for the potassium, it's an energetic favorable condition, but binding with a 6 water molecule with the sodium is its energetic favorable condition, okay? And such a binding can be kind of mimicked by the P segment in the resting potassium channel. So inside the P segment, their carboxyl oxygen of those residue, they can mimic the water molecule and bind to those potassium or sodium, okay? But the distance between these two carboxyl group, distance is perfect for the potassium, but not perfect for the sodium, okay? So as you can see here, this is a potassium ion channel. It will bind with the 8 water molecule in aqueous solution. This is a potassium energetic favorable condition. Once they approach to the P segment, those water molecule can be kind of replaced by those carboxyl group. They will be also really happy when they touch with those carboxyl group. Then they can kind of use this reaction. They can step by step and go to the other side. But if the sodium ion, in the aqueous solution, they will bind with only 6 water molecule. When they approach to this resting potassium channel, it's too small. It's only bind to the one side of the those carboxyl residue, and it cannot reach to the other side. Okay, so they will stop here. After a while, they think that the water will replace those situations because it's not an energetic favorable condition. They will go back to the aqueous solution and bind to the water. And they feel more comfortable in that situation, right? So that is why sodium will go back to the aqueous solution and cannot deliver across those resting potassium channel. Only just this difference in size can make a difference, okay? Yeah. So such a membrane potential can be measured by a method called the patch clamp, okay? So patch clamp is use some capillary tube to generate some really tiny electrode. So this electrode with a really tiny tip, the tip is only about the 0.5 micrometer diameters. And such a tip will touch the membrane and with some suction, okay, and generate such a closed environment and put some solution, okay. Then the machine can measure anything. So those are 0.5 micrometer diameter, such a tiny region only cover one or few ion channel on the plasma membrane. So anything goes through this ion channel can be measured by this machine. This is basically what the patch clamp doing, okay? So just give you this video. So this is a glass pipette with the tip is only 0.5 micrometer in diameters. It will touch to the membrane and with some suction and generate such a closed environment. Anything the ion flow will be measured by the electrode voltage difference, okay?
Thus, all the ions that flow when a single ion channel opens must flow into the pipette. The resulting electrical current, though small, can be measured with an ultra-sensitive electronic amplifier connected to the pipette. This arrangement is the cell-attached patch clamp recording method. A record of the current flowing through a single ion channel reveals when the channel is in an open or
once it's open, you will see the voltage different. This is the data come from the single ion channel. If go to the sometimes two steps, it means you are recording 2 ion channel there. Okay, so by this you can kind of you can real time measure whether those ion channel open or closed. You can add a drug, you can add a different treatment that real time measure the activity of those ion channel, whether it's open or closed. Okay, but the question is when you touch your glass pipette to the plasma membrane, how can you make sure the ion channel you are measuring is really the ion you are interested? Okay, you cannot see that, right? You just kind of just touch the member and then measure the ion flow. How can you make sure those ions are really the one you are studying? Okay, so now you need to use in that case, you need to use such a frog oocyte expression system. So frog oocyte, before fertilization, those oocytes, they are kind of in the quiescent state. They don't do anything, they just wait for sperm. Okay, so they don't, in such a situation, they don't express many ion channel or just express really low ion channel. Okay, they don't have any endogenous ion channel. So in that case, it's kind of pure system for you to do the patch clean. So you just use a glass pipette to do the microinjection to inject the mRNA which will encode the channel of your interest. Okay, so after 24 to 48 hours, those mRNA will encode the protein and it will synthesize the channel of your interest. So you kind of generate a frog oocyte which only express the channel of your interest. Then anything you measure is all come from the channel you are studying. Okay, so you need to use a patch clean and a frog oocyte expression system to achieve this. Okay, so just quickly summarize this. It's me. The potassium flow through the resting potassium channel mainly contribute to the membrane potential, okay? And also, resting potassium channel, they have a P segment, and their carboxyl residue can mimic the water molecule, and which can help potassium specificity, only deliver the potassium, but not the sodium, okay? Because its size, it's designed for the potassium, it's only good and mimic what the water molecule binding to the potassium channel, okay? They feel nothing when they approach to the potassium channel. They can kind of just behave like what they are doing in an aqueous solution, okay? And also, patch clamp can permit the measurement of the ion movement through the single channels. In the oocyte expression assay, and the patch clamp can be used to characterize the ion channel of your interest. Okay, any questions so far? No? Okay, so next we talk about the co-transporter. So there are two kinds of co-transporter, the sympater and the antipoter. So by the definition, the sympater will transport two kinds of molecules in the same direction. But the antipoter will transport two kinds of molecules in the opposite direction. Okay, so they basically will transport the one, for example, in here, they will transport the red molecule, follow its electrochemical gradient, and use this as energy to transport black molecule against its electrochemical gradient. And the antipoter will transport red molecules, follow its electrochemical gradient, and use this as the energy to transport black molecule against its electrochemical gradient, okay? But they transport in the opposite direction. So for this sympoters, we use this 2 sodium, 1 glucose sympoter as examples. So as you can see here, this is 2 sodium, 1 glucose sympoter. It contains 2 sodium binding site and 1 glucose binding site. So the 2 sodium will follow its electrochemical gradient, will bind to this binding site and then transport to the other side. So this sympater will use this as energy to also bind with the glucose and transport glucose to the other side, to against its electrochemical gradient. Okay, so the binding of those sodium basically will trigger such a conformation change, and it also have a glucose binding site. That is why once it change the conformation, it will release the glucose and finally can generate such a glucose electrochemical concentration gradient, okay? But next, you may have one question. You can tell these 2 are sodium binding site. This one is a glucose binding site, right? Why they need to simultaneously co-transport 2 molecules at the same time? Why? It look like they can transport sodium alone, okay? Or they probably cannot transport the glucose alone because it is an electrochemical gradient. But to follow the red sodium electrochemical gradient, of course they can transport sodium alone to the other side, right? It's reasonable but it's not the case. Why they call this important? They need to co-transport two kind of molecules at the same time. Why? Okay, so we use this example. The one sodium and one leucine symphoter, it need to co-transport sodium and one leucine at the same time. It must be, okay? Why? Because, as I mentioned before, the sodium would like to have a 6 water molecule to bind on their surface, right? And in this symphoter will provide 5 carboxyl group residue to mimic the water molecule, and one carboxyl residue is contribute from the leucine they co-transport. Okay, in this situation, such a sodium will feel to read, oh, this is an energetic favorable condition. Only in the case the leucine come together, okay, so they need to co-transport together. So the 5 binding site contribute from the symphoter. One come from the leucine will be co-transported, okay? So that is why they need to kind of co-transport together. Otherwise, they cannot generate such an energy favorable condition, and they cannot transport any of them alone, okay? And this is the case of the antipoters. So this is a red blood cell. Red blood cell in the different tissue, they will do the opposite function. For example, in our capillary, which is an environment with a high CO2 pressure and a low O2 pressure. So the red blood cell, they need to release the O2 and absorb more CO2 in our capillary environment. But once they go back to the lung, okay, which the environment is a low CO2 pressure and have a higher O2 pressure. So in that case, they need to release the CO2 and absorb more O2 from the lung. Okay, they need to do such a gas exchange. Okay, so how can they do this opposite reaction? Okay, so it's also contribute from this antipoter. I would say this antipoter balance such an opposite reaction. I would not say this antipoter contribute or drive the whole reaction. I would say it's only balance what happened inside the cell for those opposite reaction. Okay, so first, let us take a look here. CO2, basically CO2 is dissolved poorly in the aqueous solution. It's difficult to dissolve in the water. That is why we were burped after drinking a lot of soda, right? Because CO2 cannot dissolve in our solutions, so we burp like that, right? Okay, so the CO2 difficult to be captured, and the CO2 will be easier to permeable the plasma membrane. So the cell basically cannot capture those CO2, okay? So they need to find some way to kind of capture the CO2. So the carbonic anhydrase, this enzyme will combine the CO2 and the OH group, they will become HCO3. HCO3 will be dissolved in the solution, then it can capture HCO3, okay? And this OH group comes from the water splitting. So water will split OH and the proton, and the OH will combine CO2, become the HCO3. And this proton from water splitting will bind to, this is the hemoglobin, which is the guy can bind and release the O2. It's one kind of important protein in the red blood cell. They will bind and release O2. And then one residue, histone, on this hemoglobin, which showed here, binding with those proton will change their conformation and then will kind of release, lower their affinity with the oxygen and release the oxygen. That is why O2 will go out to the environment and provide the oxygen in our capillary, okay, in our tissue. And such accumulating the HCO3 will transport to the extracellular space, and this antipoter called AE1 will transport chloride inside the cell to balance such a reaction. Okay, but once this red blood cell go back to the lung, they will release the more CO2 here, and such HCO3 used carbonic anhydrase will split HCO3 to the CO2 and OH, and this red blood cell start burp. Okay, they release the CO2, and such OH will bind with the proton to become waters, And such a proton, the associate from the histone residue of those hemoglobin will change the conformation. Then they will have a stronger affinity with the oxygen. They will absorb more oxygen. Okay, and those HCO3 were delivered from the extracellular space by this AE1 protein, and they will also transport more chloride outside the cell. Okay, so this antipoter will balance such a reaction in the red blood cell. Okay, and also such an antipoter will also important for other reaction. For example, in the plant cell, we all know the vacuum is the important organelle to store the critical component for the plant cell, right? How can they achieve this? So first, they need to use energy. Of course, if you want to store specific molecule, you need energy. Okay, so several proton pump protein will use energy from the ATP hydrolysis to pump a lot of proton into the plant vacuum lumens. And also, it will generate electrochemical gradient, and some chloride and ion will go into the lumens, okay? And also NO3 will also go to lumen. And then it will generate a strong proton electrochemical gradient of the proton. So such an electrochemical gradient will drive those antipoter to deliver proton to the cytosol. And they use this energy to transport the sodium, calcium, sucrose, any essential molecule into store in the vacuum. Okay, that's the way the plant cells are stored, try to accumulate molecule in their vacuum. Okay, and also because the proton will bind with the chloride to form the HCl, that is why they will lower the pH level. So usually that is why the vacuum lumen is usually very acidic, like a pH 3 to 6. Okay. So in this part, we just give you an example. The sympater, which will co-transport different molecules in the same direction. Antipoter will co-transport different molecules in the opposite direction. And then for the sympater, we give you an example: 2 sodium, 1 glucose sympater co-transport sodium and glucose in the same time. And also give you kind of sodium and the 1 sodium, 1 leucine sympater to prove why they need to co-transport 2 kind of molecule at the same time. Okay, and also give you the example for the antipoter, the AE1 protein in the red blood cell, they will co-transport HCO3 and the chloride in the opposite direction to balance such reactions. And in the plant vacuum, different transport protein enable plant vacuum to accumulate the metabolites and the ion through such an antipoter. Okay, anything? No question? Yeah.
Is it correct to imagine that the sodium leucine transporter, they have the design that the 5 binding site from the transporter?
Yes.
So how about the sodium glucose sympathers? Do they have the similar design?
I guess, yes. Yeah. But people already have a clear structure information about the sodium and leucine sympater. That is why they give this as an example. I believe, I would not say that it also kind of contribute the binding site, but I would say it's the 2 co-transport molecules, they contribute to such a transport situation. Yeah. Okay? Okay, so next, in the final section of this chapter, we will give you 3 physiological, a real example, how this ion channel coordinate with each other and the trigger to kind of regulate our physiological condition. So in our stomach, okay, we eat food, and the food after digestion, those digested food will gradually go to the lumen of our intestine, right? And if you enlarge the intestine wall, you will see this is there are a lot of intestine epithelial cell on those intestine wall. This is one kind of cell, a lot of cell here. And they will try to, and the food will be here, right? The food nutrient will be here, intestine lumen. So those cells will work really hard to absorb the nutrients from the lumen of the intestine and try to deliver those nutrients to the lower area, which is the circulation system. They will deliver nutrient to the circulation system, and through the circulation system, those nutrients will go to the other part of our body. That is why we can get energy. Other part of our tissue can get energy from the food. Okay, so if you enlarge the cell here, this is one kind of intensity epithelial cell, their neighbor cells. Okay, they need this is the lumen site, and this is the circulation site. They need to absorb the nutrient from this side and pump the nutrient to the circulation site. Okay, that's their goal. Okay, so how can they do this? So in the beginning, there's a sodium potassium ATP power pump. They will use energy from the ATP hydrolysis to transport a lot of sodium to this side and the potassium to the other side, and then generate the electrochemical gradient like this. And then this symporter will use sodium, will transport sodium, follow its electrochemical gradient, then use this as energy to transport more glucose inside the cell. Accumulate more glucose, then this group 1 is a unipolar. It will transport glucose from the cytosol to the other side. Okay, follow is an electrochemical gradient. Then they can deliver, their main goal is deliver the glucose from this side to the other side. And glucose nutrient will go to the circulation system and go to the other part of our body. Okay? So they need the energy to do this. Okay? And also the second example, how our stomach lumen is really acidic, right? It's really good for some enzyme reaction and help to digest the food. Okay, how such a stomach can generate such a super acidic environment? So it will contribute from the cell on the stomach wall. Okay, so this is stomach lumen. This is the cell on the stomach wall. So there are a lot of epithelial cells. They use those proton potassium ATPase. They use energy from the ATP hydrolysis to transport a lot of proton and then generate such an electrochemical gradient. So those proton will go back here, and then they use this potassium will go back here. The proton will transport to the other side. So they will accumulate more proton here. And this proton comes from the H water splitting, and the OH will bind to the CO2, become the HCO3. So this chloride HCO3 antipolar will use this HCO3, transport this HCO3 as an energy to co-transport chloride into the cell. And then this chloride will deliver it through the chloride channel and to the stomach lumen. So combine those proton and the chloride, they will become HCl and lower the pH level of the stomach lumen. Okay, so the energy will come from this proton potassium ATPase. The final example, two kind of cells. One is this, the word is, the form is small, osteoblast and osteoclast. Two kind of cell. Those cells are really critical for our bone. Okay, so osteoblast, they can kind of generate more bone. Osteoclast will eat bone, will digest bone. The coordination between these two kind of cells, they can shape the normal pattern or normal structure of our bone. Okay, so we will focus on here how such an osteoclast bind to the bone and digest the bone. So if you enlarge here, you will see this is the osteoclast. The bottom will be the bone. They will bind to the bone and make a really tight seal between the bone, and it will generate a closed environment, closed lumen here, and they will try to make this lumen really acidic because several enzymes required for such a bone digestion need an acidic environment. Okay, so they will use this proton pump, use energy from the ATP hydrolysis to transport a lot of proton to this side. This proton comes from the water splitting. It's really similar to mechanism to the red blood cell and the stomach cell, right? So the OH will bind with the CO2 and become the HCO3, and the antipolar will deliver the chloride, and such a chloride channel will deliver chloride to this closed environment. So combination of the H proton and the chloride will become the HCl and become really acidic here. So the enzyme here will have a better enzyme reaction and try to digest the bone here. So they can kind of really locally and precisely digest the bone. They will not destroy the whole bone. Okay, so through such a precision, they can shape the bone to the normal structure. Okay, so basically these will be the three a simple example to show you the coordination between the different ion channel, importer, or ATP power pump. They were kind of really important to trigger the normal physiological condition. Okay,